Hello viewers, welcome to my channel. Today we will discuss on microbiology question paper of GATE Excel 2022. In my previous video, I have already discussed on some question of GATE Excel Microbiology 2022. The remaining part today we will discuss. Some brief notes regarding the question and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. What will be the value of the numerical aperture if the half aperture angle is 58 degree and the oil immersed objective is used for the process of light microscopy? Consider sin 58 degree is equal to 0.85 and the refractive index of immersion oil used is 1.50. To answer this question, we have to consider the formula Na is equal to small n sin theta, where Na is equal to numerical aperture, n is the refractive index, theta is half aperture angle, that is 58 degree. Here sin 58 degree is equal to 0.85 and the refractive index of immersion oil used is 1.50. So the numerical aperture Na is equal to 1.50 into 0.85, that is 1.275. As we have to answer up to one decimal place, 1.275 is approximately 1.3. So the answer will be 1.3. Now let's discuss about the refractive index and numerical aperture. The refractive index is a measure of bending of a light ray when passing from one medium to another, whereas the numerical aperture is the number associated with the light gathering capacity of an objective lens or condenser lens. Next question. For this question, there are 4 options mentioned here. And you have to choose the correct sequence of overall biochemical reaction which express the process of denitrification. Denitrification is a microbially facilitated process where nitrate is reduced and ultimately produces molecular nitrogen through a series of intermediate gaseous nitrogen oxide products. This process is performed primarily by heterotrophic bacteria such as Paracoccus denitrificans and various pseudomonas. The sequence of reaction is from nitrate, it is from nitrite, that is NO2, to nitric oxide, that is NO. This nitric oxide then converted to nitrous oxide, that is N2O. Finally, this nitrous oxide is converted to dinitrogen, that is gaseous nitrogen. So the correct answer will be the option B. Next question. Escherichia coli, that is E. coli, growing under favorable condition doubles in every 20 minutes. If the initial number of E. coli cell is 100, what will be the logarithmic number of cells at 17th generation? To solve this question, we have to consider the formula n is equal to log nt minus log n0 divided by 0.301, where n0 is the initial population number of bacteria, nt is the number of population at time t, and n is the number of generation. Here the initial number of E. coli cell is 100 and we have to calculate the logarithmic number of cells at 17th generation. So the number of generation here is 17. So number of generation n is equal to 17 and the initial number of bacteria, that is n0, is equal to 100. That is n is equal to log nt minus log 100 divided by 0.301. That is 17 is equal to log nt minus log 10 to the power 2 divided by 0.301. Finally, log nt is equal to 7.11. As log 10 to the power 2 is equal to 2 into log 10 base 10, that is 2. So log 10 to the power 2 means 2. So the correct answer will be the 7.1. because we have to answer up to one decimal place. So 7.11 is approximately 7.1. If you want to learn more about bacterial growth related mathematical problems, I have already made a video on this. Link is available in the description box. Next question. Which of the following methods are used for the identification of microorganism? Option A is nucleic acid hybridization. Option B is Southern blotting. Option C is 16S rRNA sequencing. And option D is percentage of GC content, which determine the melting temperature. And the correct answer will be either nucleic acid hybridization or 16S rRNA sequencing or Southern blotting. These three options are correct. So this is MSQ type question because multiple options are correct here. Nucleic acid hybridization is useful for differentiated two bacteria. DNA-DNA hybridization is useful to study only closely related bacteria, whereas the DNA-RNA hybridization helps comparing distantly related bacteria. Nucleic acid, that is DNA and RNA sequencing such as 5S and 16S rRNAs directly compare the genomic structure, thus identify the bacteria. rRNA are almost ideal for the studies of bacterial, that is microbial evolution and relatedness. GC ratio is also helpful in characterizing bacterial genera. For example, Staphylococcus and Micrococcus are genera of gram-positive cocci, having many features in common, but they differ in their GC ratio by more than 10%. The former, that is the Staphylococcus, has a GC ratio of 30 to 38%, whereas the latter, that is the Micrococcus, has a GC ratio 64 to 75%. Although the GC ratio is helpful in characterizing the bacterial genera, but the GC ratio of strains within a particular species is constant. So it cannot differentiate between the strains within a particular species. Southern blot is a method useful in molecular biology for detection of a specific DNA sequence in DNA samples. Southern blotting combines transfer of electrophoresis separated DNA fragments to a filter membrane and subsequent fragment detection by probe hybridization. The primary uses of Southern blotting is to identify a specific DNA in a DNA sample. It mostly used in the identification of viral infection and subtle bacterial infection. In recombinant DNA technology, Southern blotting technique is used to isolate particular DNA. Next question. If the plasmid DNA given here is digested with restriction enzymes HINT3 and EcoR1, considering complete digestion, how many DNA fragments will be formed? Here it is mentioned that HINT3 and EcoR1 both causes complete digestion of this DNA fragment. So here the fragment A, B, C, D will be formed. So the number of fragments released will be 4. Next question. Which of the following are present in gram-negative bacteria? Option A is lipopolysaccharide. Option B is teichoic acid. Option C is periplasm. And option D is endotoxin. And the correct answer is both lipopolysaccharide or endotoxin. So both options are correct. So this is also a MSQ type question. Teichoic acid are present in gram-positive bacteria, whereas lipopolysaccharide and endotoxin are present in gram-negative bacteria. Gram-negative bacteria are surrounded by a thin peptidoglycan layer, which itself is surrounded by an outer membrane containing lipopolysaccharide. Today, the term endotoxin is often used synonymously with LPS. Bacterial endotoxin found in the outer membrane of gram-negative bacteria are members of a class of phospholipid called lipopolysaccharide. Endotoxin is a type of pyrogen. LPS are not exogenous product of gram-negative bacteria. The release of LPS from bacteria takes place after death and lysis of the cell. Next question. Match the correct mode of cell division with respective bacteria. Here 4 bacteria is mentioned. P is Streptomyces species, Q Rhodopseudomonas acidophylla, R Bacillus subtilis, S is Nocardia species. And also the 4 mode of cell division is also mentioned. In option 1, here it is found that septon or crosswall is formed at the middle of the cell, and it causes production of 2 equal size of cell. In option 2, it is also found that a small bud or protuberance is formed at the end of the cell that eventually enlarge and separate and form 2 cells. In the third option, the cell causes irregular partitioning and produces 3 cells. And in option 4, the newly formed cells are attached to each other. Here the option will be the option C, that is P is equal to 4. P means Streptomyces species is option 4. For Q, that is for Rhodopseudomonas acidophylla, it is option 2. For R, that is Bacillus subtilis, it is option 1. And the remaining is Nocardia species, it is S is 3. The mode of cell division of these 4 bacteria is mentioned here. The gram-positive bacterium Bacillus subtilis are divided via 2 modes of cell division. During vegetative growth, the division septum is formed at the mid cell to produce 2 equal daughter cells. That is in option 1, the division septum is formed at the middle of the cells and produce 2 equal cells. So for Bacillus subtilis, R is 1. Here also in option C, R is 1. Next one is the filamentous actinobacteria. Streptomyces undergo 2 distinct modes of cell division, leading to partitioning of growing hyphae into multicellular compartment via crosswall at irregular interval. The individual filaments remain physically connected. That is for Rhodopseudomonas acidophylla. For Streptomyces is P is equal to 4, P4. 4, that is individual cells are connected or attached to each other. So for P, that is Streptomyces species, it is option 4. Here in the answer P is option 4 is mentioned. Next is Rhodopseudomonas acidophylla, that reproduce by budding, a process in which a small protuberance or bud develops at one end of the cells, and this enlarges and eventually develop into a new cell which separates from the parent. That is, bud develops at one end of the cells. In option 2, bud develops at one end of the cells that enlarges and eventually produce 2 cells. That is for Q option, that is Rhodopseudomonas acidophyllus. The option is 2. That is for Q, it is 2. In the option also, the Q is 2. And the last is the Nocardia species. The option 3 is remain. So for Nocardia species or S, it is 3. Nocardia species are aerobic actinomycetes that reproduce by fragmenting into bacillary and coccoid elements, distinguished by filamentous growth with true branching. Thank you. Please like, share and subscribe to this channel. Thank you.